Okay, let's dive right into this. Welcome to today's Explainer, where we're opening up a true medical mystery box at the very cutting edge of cancer research. We're tackling one of the absolute biggest headaches in oncology today, acquired resistance to immunotherapy. Just imagine the scenario for a second. A patient gets a breakthrough treatment, the tumor visibly shrinks, their immune system is finally fighting back and winning, and then, out of nowhere, the cancer finds a loophole. It escapes. So how on earth does it do that? And more importantly, how do we shut that escape route down for good?
That is the massive million-dollar question we're exploring today. Immune checkpoint blockade, or immunotherapy, has completely revolutionized cancer care, basically by taking the brakes off a patient's own immune system. But cancer is a ruthless, brilliant biological escapologist. Even when these therapies work like magic at first, a solid chunk of tumors eventually find a way around it. They adapt, they evolve, and they go right back on the attack. To figure out exactly how they pull off this disappearing act, we're going to dig into an enormous new pan-cancer study that finally pulls back the curtain on this exact phenomenon.
So here's our game plan for today. We'll start with the immunotherapy catch, then look at a massive investigation. After that, we've got the inflamed twist, followed by unmasking the culprits. Then we'll weigh the strengths and limitations and wrap it all up with the future of cancer care.
All right, part one, the immunotherapy catch. Let's lay out exactly what we're up against.
The specific clinical phenomenon we're zeroing in on here is called acquired resistance. Now, for this study, researchers define this using very strict real-world criteria. It's when a patient's cancer actually responds to immunotherapy, meaning they have durable, stable disease for at least 6 months, but then eventually it progresses again. The treatment was absolutely working, but the tumor figured out a workaround. And get this, the data shows this happens in roughly 22 to 34% of treated patients. The rates are actually even higher for folks getting combination chemoimmunotherapy compared to just targeted monotherapy. And this brilliantly illustrates an absolutely vital distinction in oncology. Acquired resistance means the therapy did work initially, but the tumor actively circumvented it. Primary resistance, on the other hand, means the immunotherapy literally never worked from day one. The tumor was completely unfazed. Why is that distinction such a big deal? Well, because these two scenarios are driven by entirely different evolutionary and immunological mechanisms. Grouping them together, which we've kind of done in the past, just muddies the waters. But this study carefully separates them so we can see exactly what happens under the pressure of a successful initial treatment.
Moving on to Section 2, a massive real-world investigation. It's time for some detective work.
Okay, consider this massive number, 3,559. That is the number of immunotherapy-treated patients included in this cohort. It's truly staggering. See, older studies on this topic usually relied on really small groups, or they only looked at melanoma or just lung cancer. But this team totally flipped a script, utilizing a huge real-world clinicogenomic dataset spanning multiple cancer types. We're talking non-small cell lung cancer, head and neck squamous cell carcinoma, and triple-negative breast cancer. Oh, and to make absolutely sure their findings were actually specific to immune checkpoint blockade, they threw in nearly 1,900 non-immunotherapy control patients as well.
Because they had this unbelievable scale to work with, the researchers spotted something totally wild in the survival data. If you look at post-progression survival, basically how long patients lived after the tumor started growing again, patients with acquired resistance actually survived longer than those with primary resistance. But here's the real kicker. This survival advantage was unique to immunotherapy. When they looked at the control groups, the folks getting traditional chemo or targeted TKI therapies, that advantage just vanished. So what does this tell us? It strongly suggests that a successful initial immune response gives the patient a lasting clinical benefit, even after the tumor technically breaks loose.
Which brings us to Section 3, the inflamed plot twist. This is where things get really interesting in the tumor microenvironment.
The biological plot twist here is just fascinating. Honestly, the logical assumption would be that an escaped tumor just hides, right? Like it turns immunologically cold and totally shuts down the immune response. But nope, the data showed the exact opposite. Acquired resistant tumors actually remained highly immune inflamed. They had even more immune cell infiltration and inflammatory signaling than the primary resistant tumors. But there's a huge catch. The inflammation was completely dysfunctional. These tricky tumors were actively using alternative checkpoints and immunosuppressive pathways to survive right in the middle of all that persistent inflammation. So the immune system is still violently knocking at the front door, but the tumor just changed the locks.
Let's head into Section 4, unmasking the genetic culprits. It's time to find out exactly how they pull off this escape. To track down the specific mutations that give these tumors their get-out-of-jail-free card, the researchers didn't just round up the usual suspects. They used a heavy-duty analytical tool called DN/DS analysis. Now, without getting completely lost in the math here, this is essentially an unbiased method that tests whether genetic mutations are undergoing positive selection compared to the normal expected background mutation rate. In plain English, it proves that these genetic changes aren't just random noise or some lucky byproduct of a growing tumor. No, they were actively and specifically selected by the intense evolutionary pressure of the immunotherapy itself.
So, the crucial point is, what did they actually find? Well, they definitely confirmed some known drivers, like B2M loss of function in non-small cell lung cancer, which was great because it validated their whole approach. But then they uncovered this entire suite of novel culprits. We're talking TGF-BR2 and ARID1B in lung cancer, CYLD and FAT1 in head and neck cancer, and RUNX1 in triple-negative breast cancer. And the major takeaway here is that these different mutations create entirely distinct escape phenotypes. Acquired resistance isn't just one single biological state. Depending on its specific mutation, a tumor might use immune exclusion, or reprogram its metabolism, or trigger suppressive myeloid cell infiltration just to stay alive.
Moving on to Section 5, Strengths and Limitations. Let's take a step back and objectively evaluate the evidence here.
Now, the strengths of this study are frankly monumental. The sheer real-world scale, the inclusion of non-immunotherapy controls, and that sharp focus on post-progression tumor sampling, it makes this the first broad pan-cancer multimodal comparison of its kind. But, it does have a few blind spots. The biggest limitation is that the pre-treatment and post-treatment biopsies mostly weren't patient-matched, meaning they came from different people, so we can't definitively trace the evolution within a single individual patient. On top of that, they used targeted DNA panels, which can sometimes miss larger structural rearrangements, and they used bulk RNA sequencing. Bulk sequencing is great, but unfortunately it lacks that ultra-fine cell-by-cell spatial resolution we really need to see exactly how the immune and tumor cells are physically interacting on the battlefield.
Finally, we arrive at Section 6, the future of cancer care. How does all this data pave the way for tomorrow's treatments? Let's move to the next logical step and see how this builds a concrete pipeline for future research. Step 1 is definitely prioritizing prospective longitudinal studies with matched biopsies, actually tracking the exact same patient's tumor over time. Step 2 is moving beyond that bulk data into single-cell profiling and, critically, utilizing serial liquid biopsies like circulating tumor DNA. Just imagine the power of being able to monitor resistance mutations like B2M or RUNX1 in real time, just from a simple blood draw, long before the tumor ever shows up growing on an X-ray. And that perfectly sets up Step 3, biomarker-guided clinical trials. Instead of treating every patient who fails immunotherapy with the exact same backup plan, we can match their specific genetic escape mechanism to a brand new, highly targeted therapy.
Which leaves us with this final, truly provocative thought. If we can accurately map out exactly how a tumor escapes, whether it's hacking a metabolic pathway or upregulating some alternative immune checkpoint, can we turn acquired resistance from a devastating treatment failure into a personalized roadmap for a cure? This study really suggests that we are standing on the brink of an era where a tumor's greatest escape trick might actually become the exact thing that tells us how to defeat it for good. Thank you so much for joining me for this explainer, and I really hope this deep dive sparked your curiosity about the incredible future of individualized cancer care. Catch you next time.